Everyone. So my name is Cheng, and we're going to talk about our research that have been really going on for the past few years on self-supervised methods to learn representations for host-like images. Right, so I'll start with a brief background, and then I'm going to talk about a few different methods that we're introducing, and then we'll wrap up quickly with the conclusion. Right, so let's dive into the background. What we're interested in here is biomedical microscopy. Biomedical microscopy is a very important imaging method and diagnostic tool for both research and clinical medicine. Biomedical microscopy can help us to do a tumor diagnosis, molecular classification. It can help us predict a patient's outcome, right, or predict how a patient might react to some treatment, right? And this is also very important for use in clinical trials, enrollment, or for clinical trial enrollment, or maybe even formulate a surgical plan for these patients who maybe have a tumor, right? So more recently, digital pathology and host-like imaging really increased the role of computer vision and machine learning in analyzing these microscopy data, which is very complex and very large, right? So there are two types of imaging methods that we're interested in. The first one is H&E staining, right? H&E staining has been the state-of-the-art and standard of care for probably more than 100 years or so, right? This is the kind of images that pathologists are very familiar with, and they're trained with that, and they kind of use that to diagnose patients. And that's the standard of care right now. And the other modality that we're interested in is SRH, right? So stimulated Raman histology. And SRH is a relatively more novel way to image these specimens using optical imaging. And the upside for SRH is that we don't really need to process the tissues, right? We can image these specimens fresh. So we don't need to do staining, we don't need to do fixing. And the upside for this is that we can do this within minutes. So we can do this right in the OR where we can have a specimen coming from the patient and goes directly into the microscope, and we can see the image right away. And that's why we like SRH so much, because we can get this image very rapidly, and we can have some diagnosis right there to maybe help the surgeon to figure out their treatment plan going forward. And this is where AI has a very large role in formulating how these, in looking at how this data is being treated.
Sorry, just a quick question. So I can see a lot of advantages of SHR over HID,
Yeah.
but in terms of resolution, which one is better?
Yeah, so the question is, what's the resolution wise, how does SRH compare to H&E? So H&E actually has, so if we're talking about like magnification of the specimens, SRH is usually being done at 20x, and roughly. And H&E can be done at 20x if you need it quickly for diagnosis, and it can be done up to 40x. The vast majority of the data that we have are when we are actually processing these data is done at a scale of 20x. So roughly there is, in terms of the resolution, they're pretty similar. But SRH also have another big advantage in the sense that the data is very standardized. We don't really need to rely on maybe technicians' experience, or maybe they overstain something or understain something, or there's no variability between different institutions. Right? SRH is a very standard machine, a very standard microscope, right? So we have the tissue in, and it doesn't matter which microscope it is, the image will always look the same. Yep. So these are the two microscopy modalities that we're working with. And these images are actually very big. So I'll give you an example. These are basically two host-like images of H&E and SRH. So SRH is a bit smaller than the H&E because it's a more focused window, right? We already have a window region where a lot of this white space here on the H&E image really isn't important. We kind of just throw them out when we're processing them. So that's why there's kind of like this size difference. But if we look at this difference if we look at these sizes in terms of megabytes, right? A H&E whole slide image can go up to 70 gigabytes, right? And a SRH image can go up to 600 to maybe a gigabyte, right? And these images are just so big that it's really challenging to process them with deep learning, right? And especially if we want a batch size of, let's say, 1024, right? There's not enough GPU, there's no GPU on the market that can actually process these images, right? And just for comparison, if we look at natural images on the ImageNet, they're usually 256 by 256, and they're only a few hundred kilobytes, right? So this big difference is one of the most important challenges that come with histology or whole slide images. So what a lot of prior work has been done is what we do is we essentially patch these whole slide image into smaller patches, right? So that's a solution to handle this huge size, right? So when we have a whole slide image, we will patch them into smaller sizes, right? And these sizes are maybe 256 by 256 or something similar to that, that we can easily digest those sizes with modern techniques. And then we will use a patch encoder to patch to encode each patches, right? And then from each patch, we'll have some kind of patch token to represent these patches. And once we have that, we will then use a slide encoder that takes in the output of our patch encoder, right? We'll go from the patch tokens to some kind of slide embedding, right? So we have this two-stage approach. This approach can also be called multiple instance learning or MIL approach, but this is has been an established pipeline that how we have been processing these data. Another challenge that come with these images is that all the labels that we have about these data is really at the patient or host slide level, right? So if we're lucky, maybe we have a pathology report, right? In this pathology report, a trained pathologist will describe what they see on the images, or maybe they see a few different attributes on the image, but they're not really anchored to any point, right, on the image. They'll just see that we see some attributes on this image, but they don't necessarily tell you where. And the vast majority of the cases that we have are actually, we just have a table of the labels that we have, right? We know this is a high-grade glioma, that's great with some molecular information that's IDH wild type, right? So these very high-level information, these labels about these data is really coarse, and it's really at the patient level, right? So, and this make it quite challenging for us to actually train a model to process them, right? So to summarize the challenges that we have with these whole slide images, we have very large whole slide images, right? The annotations that we have is weak labels. It's only at the slide or the patient level. And these fine-grained labels is really, really challenging to acquire, right? We are not going to sit a pathologist in a room and say, here's 1 million patches and you have to label them, right? Tell us this is tumor, this is blood, this is necrosis. That's just not feasible for us to do. And even so, even if we can do that, right? The pathologist can only give us histologic annotations, right? The pathologist can see, I see some infiltration here, I see normal tissue, or I see some blood, I see some necrosis. These very high-level descriptions of what we can see on the image, which is good to have, but it's really not the most interesting thing that we want to do, which is maybe classification on the molecular scale, or maybe how the patient will respond to a treatment, or how long the patient will survive, right? So the pathologist doesn't necessarily have a globe that can tell in the future. So because of these challenges, there is a need for having a self-supervised representation learning methods for histopathology data. So here, self-supervised means that we want to learn good representation without actually utilizing these labels that we have for the patients, right? We'll use these labels to evaluate how the model is doing, But the goal here is to, when we are training these models, right, we don't want to actually use any of these labels because we know we don't have a lot of labels. They're not very good, or they're very sparse. So, so in today's talk, we're going to talk about our proposals, right? Our vision on how to train a histopathology, a whole slide image representation learning, right? With a patch encoder and a slide encoder in this framework, right? So, and I will start with the patch encoder on how we are going to use SSL to better train these patch encoders, right? And so, so our way, the way that we're proposing to train the patch encoder is hierarchical discrimination, high disk. So I'll give you a little bit of a background on how existing self-supervised learning work for patches, right? So we have these patches, right? We have a whole slide image. We have already kind of divided them up into smaller patches. So we have this patch which is very nicely sized. We can fit that onto the GPU. What happens is we actually do different transformations to this same patch, right? So the idea here is that maybe similar patches should have similar representations, right? So how do we get this similar patches, right? Because we're kind of assuming that this patch is all we have, right? So the easiest way that a lot of computer vision researchers do is to essentially apply different transformations on the same image. And maybe we'll flip it around, maybe we'll blur the image, maybe we'll add some noise, maybe we can change the color, change the contrast, make it more bluish, make it more reddish, things like that. And the idea here is that we want to encourage the model to know that these transformations that we have applied for on these images are really not important, right? These images are still similar, and therefore they should have similar representations. So once we have these transformations, we pass them through a neural network. And there's a lot of different research on this, how this neural network should work. Maybe there's some kind of weight sharing between the two neural networks. So it might be the same neural network, or maybe one neural network has a few more layers. Maybe one neural network is not being updated. There's a lot of different research in this area, but the vast majority of them do share this common architecture where there is this, what we call a Siamese network, right? We have two networks that could be the same to extract features from each of our transformed image. And from there, the idea here is that these transformed images should be similar. So some kind of loss function is being employed to make sure that these images will have similar representations, while all the other images, right? So maybe we train our model with a maybe batch size of 512 or 1024, then all the other images are not the same image, right? They're not the same image that we have transformed. So they're what we call negative pairs, right? And the two images that we transform are the positive pairs. So the idea here is to minimize the difference between positive pairs and maximize the difference between positive and the negative pairs. All right, does that make sense to everyone? Yeah, great. So this is kind of what people have been doing for a while, and what we see here is that there's really a lot of disadvantage of using this technique to learn these representations. They do a good job for a lot of tasks, right? But how can we push the performance of this training paradigm further, right? So a few different weaknesses of this approach that we have seen is that it assumes the patch independence, right? So in our mini batch, right, in our batch of maybe 512 images that we're looking at, we're assuming that every patch is independent, where that may not necessarily be the case, right? So for example, if we sampled multiple images from the same patient, right, they're definitely not independent, right? We actually have a pretty good idea that maybe they should also be quite similar to each other. Where in patch instance discrimination, these will actually be considered negative pairs, right? Because we're kind of not treating, we're trying to do this without seeing any labels. This approach actually also neglect this hierarchy that we have of our data, right? Because in our histologic data, there's a natural hierarchy that goes from patient to slide and then to patches, right? So a patient may have a few different slides, and every slide may have a few thousand patches, right? So this hierarchy is not really taken advantage of, right? Because we know this hierarchy give us some information on how these data might be organized. And finally, this really requires strong data augmentations. That's really not very informative and it's domain agnostic, right? We're training these data in the same way that we train a data set on the ImageNet, right? So sometimes these augmentations can even corrupt semantically important features on the image and maybe have us learn something that's really not important. So to combat these like weaknesses of the prior work, we propose HIDISC. Right, so this is kind of like the overall of what HIDISC do, and I'm going to focus on just at the bottom first, right? So what we have here at the bottom of the hierarchy is that is basically what we have before, right? We have different patches and we have the patch and the transformation, they're positive pairs with each other, right? And in this hierarchy, these two different views are the positive pairs and everything else will be considered as negative pairs, right? So that's what we had before. And this is what we call patch discrimination, right? We're discriminating the instances at the patch level. So what we can do is actually look at where these patches come from, right? Maybe these patches come from the same slide, right? And if we have this information, which we almost always have, we can then construct positive pairs based on their slide membership, right? So maybe these two patches are from the same host slide image, and then they will be considered a positive pair, right? They look very different, right? Compared to two different augmentations of the same image. But the idea here is that they encode the same underlying tumor biology, that they actually show us the same thing, right? So that's what we call slide discrimination. We're doing instance discrimination again, but only at the slide level. And finally, we can do this at the patient level as well, where maybe we do know that maybe a patient had multiple specimens, and these specimens are again the same views of the same underlying tumor biology. So we can also include maybe patch here and a patch here as a positive pair if they come from the same patient, right? So this is how we can essentially utilize this very large image size to help us find very natural augmentations just because they're different views of the same tumor, right? Instead of having to handcraft them or change the color or flip them. And the idea here is that if we use slide discrimination to train a patch encoder, our patch encoder can actually learn slide level information, right? These representations will actually tell us what is happening at the slide level. And likewise, if we do that at the patient level, right, our patch encoder have the information of this patient already, right? So we think this is a very strong way to do these to avoid these transformations and give us better representations for these patches, right? So this is a little technical here. I'm just gonna quickly dive into pseudocode for training our method. This is quite similar to how most method is being trained. Here what we do here is we compute the same loss function 3 times, right? So we can compute the loss function based on the patches, right? And then this is just the patch discrimination. And we can compute the loss at the slide level, and then that gives us slide discrimination. And we also compute the loss at the patient level, right? So we can add these loss functions together to kind of balance out right, the different ways that we want to weight maybe patch discrimination, or maybe we want to weight slide discrimination more to kind of have a better custom representation for maybe a different task that we're interested in. Going a little more into the representation. going into the loss function, right? So the loss function, so we're employing a contrastive learning objective here, which we don't have to use contrastive loss, but that's the loss that we kind of studied when we were developing this method, right? So in the contrastive loss, we kind of have the positive pair feature similarity in the numerator and the negative pairs feature similarity in the denominator. And that's kind of the similarities that we're trying to contrast. And we're doing that for all the other images in our mini batch. And tau here is a kind of temperature hyperparameter. So this is the standard, pretty standard, well-studied contrastive learning formula. But what we do here is we compute this loss over all the positive pairs at a certain level of discrimination, right? Either patch, sled, or patient discrimination. And once we do that, we can do that for all the images in our mini batch, right? So basically, for every image in our mini batch, we will find all of the positive pairs at a certain level of discrimination, and we will compute this contrastive loss. And so in this level, this loss can be computed per level of discrimination as well. So maybe at training time, we'll first find all the positive pairs at for patch discrimination, which is just the patch itself and then the augmentations, right? We'll compute the loss with these positive pairs that we identified. And then we will do the same loss function, but we'll with a different set of positive pairs that is defined by all the other patches from the same post-slide image. And then we can do the loss again with all the patches from the same patient. So this is how we formulate our loss function, and we have some weighting hyperparameters to help us weight these loss functions, and we do that for all the different levels. So we benchmarked our method on a few different data sets, including SRH and H&E data. So SRH is a 7-class classification method where we're basically trying to determine the tumor type, the histologic tumor type of different images. And on the TCGA side, we are doing a more advanced classification on molecular information, where we're trying to differentiate IDH wild type and white IDH mutant type of tumor. And the result is overwhelmingly amazing for the methods that we're proposing. And I don't want to pay too much attention to other results, but I can tell you that high-disk slide and high-disk patient completely outperformed all the baselines that we have tried by a very wide margin as well. And here, high-disk patch, right? So if we only use patch discrimination, it's actually very similar to the baselines that the prior studies that we had, and that's also something that we have seen. So high-disk patch and Sinclair are the baselines that we're comparing against, and as expected, they're very similar to each other. We also visualize these representations using t-SNE to do a dimensionality reduction on the learned representations. And what we can see here is that both high-disk and Sinclair do pretty good in terms of learning a good representation, but high-disk is better in the sense that different tumor types are better separated. And we can also actually see subclusters here within different tumor types, right? And then these subclusters actually do correspond to different patients, right? And this is not something that we are asking the model to do, right? This is just the objective that we're enforcing when we're training the model. And as we can see, this is very good in terms of having a better representation. And what's more interesting is that this patient cluster is not seen for normal patients, right? Because they're all views of the normal brain, which doesn't necessarily have differences between different patients. One of the things I mentioned earlier is that all the prior work requires strong augmentations for the different require these augmentations to form the positive pairs. So what happens if we don't have these strong augmentations, right? What we can see is that prior work and also high-disk patch, right? Remember, high-disk patch is also only patch discrimination. These models essentially just collapse. right? They can't learn anything because there is no transformation that's being applied to these patches. But as what we expected, high-risk slide and patients still perform really well, even without these domain agnostic transformations. So it still performs really well without these transformations, which is a very good strength of our method because these transformations are sometimes very compute intensive as well, and that can kind of slow down the training of these methods. And we also know that they don't necessarily correlate to any biomedical process. So to visualize the different representations, without strong augmentations, the representation of Sinclair is essentially random. So to kind of summarize our contributions in HiDisk, the HiDisk is here to leverage the inherent patient slide and patient hierarchy for biomedical microscopy representation learning. HiDisk outperform all the previous self-supervised methods for cancer diagnosis and molecular mutation prediction. And HiDisk learns very good representations for our patches, even without using the strong augmentations that's being required by all the prior works. So going back to our vision on how to train these two-stage networks, right? We believe the way to train patch encoder is through HiDisk, right? It learns very good representations and is very efficient. It doesn't require the strong augmentations that everything else requires. So to tell you more about the sliding coders, I'm going to pass it off to Renly. Yeah. Yeah, before that, I'll take questions.
Yeah, so what exactly is the network that you're using for the forward passing?
So for these experiments is ResNet 50.
ResNet 50.
That's right.
And are you using the pre-trained one from ImageNet and then, or are you training from scratch?
For these experiments, they're trained from scratch, but there's no requirement to train from scratch. It will work with ImageNet pre-training, it will work with different model architectures. But for the sake of these experiments, that's what we did.
Okay, and are you still using the Siamese setup or are you dispensing with that?
It's still Siamese setup, yeah. But the way it's shared between the two branches of the network, so it's one set of weights. Yeah.
It's the same set of weights.
It's the same set of weights, yes. It's still Siamese.
Okay, so are you freezing any of the layers during like
No.
interest? No.
No.
Okay, thank you.
Yeah, we have freezing coming up.
Oh, okay.
Yep.
How did you handle the situation where deeper resolution from deeper system? You can think of deeper resolution of the H&E information. How do we handle that?
Yeah, so the way these H&E images being stored is they kind of store everything at the highest resolution that we have. And we can kind of scale the image to a standard resolution, and that resolution is 20x. Right, so most data that we have is either 20x or 40x. Right, so for the 40x images, we basically take basically double the field of view and we just resize it down. Okay.
All right. Since for this two-stage network, the HiDisk part actually will enable us to know a very good patch representation. But the ultimate goal of the CPath or the computational pathology is to having a good slide representation, because that is part that most of the pathologies or the actual annotation or the evaluation can be done. So here, the next stage is after we get this very high quality patch tokens from the patch encoder, how do we actually have a slide encoder can first of all aggregate all those patch tokens into one slide features, and also potentially we can having a task agnostic slide encoder, which is not specifically trained to any of the tasks or something. So here is what we propose. We call it slide pre-trained transformers, or abbreviate as SPT. So the advantage of having SPT is some of the slide encoder, you do need supervised whole slide learning to this, having the annotation like the tumor type, or the IDH status or MGMT, some of these molecular informations. And this is very easily to having a task specific overfitting on the task we are going to have. And here we're proposing to have this slide encoder to learn this inherent post-slide structures with the transformation into itself, and just without any reliance on those tasks or these dense annotations. Similar as before, here we have the whole slide imaging or patch name. We put them into the patch encoder, which is already trained is pretty well. It's doing pretty well, and also we'll apply these two different post slide transformations to generate the two views for the same post slide images. That is where our like either contrastive view or the Siamese two different Siamese views formulated. And then we'll put them into the same. Actually, we'll put two here, but they're shared with our whole slide transformers. And they will also only get one token. Usually we'll do a class token out of it. And then we can apply any like self-supervised learning method. Like we talked about before, we can have like SimClear, we can have BYOL, we can have BigCrack, or even we can use some supervised method that requires like two different views to do it. For example, SupCom, they integrate both SimClear and also the label information for the supervised loss. That is part we cluster we classify our SPT into two ways. One is self-supervised SPT, one is supervised SPT. We call it SS SPT and SU SPT. Okay, so the very important or key ingredient in our SPT is how do we generate two different views for the same post-slide images. For the whole slide images, there are lots of features, and also it's actually quite challenging to do it. So we can have a regional heterogeneity. We'll have very mix of the dense tumor. We have the normal tissue. We have some blood cell and maybe some necrotic regions. They are very different in the same post-slide images. So we want to handle these challenges. And even within the same type of the tumor, well, we maybe have quite different histological features to handle. And also is the common issue for the computer vision, and is even worse situation for the whole slide images is information redundancy, where for this gigascale pixel images, you usually don't need to have every patch token or everything, but how do we kind of handle this information redundancy to reduce them to very dense representation is ultimate goal to formulate a very high quality slide encoders. Here we propose three different ways to handle them. First is splitting, and then it's cropping, and the last one is masking. So the splitting is we designed specifically to decrease the mutual information between two different views, because as we have shown in the like previous high dispatch augmentation, if we just apply patch, basically two views will have exactly similar information. In that way, your self-supervised learning objective does not provide very beneficial or any structure that they can learn. So the splitting is we don't allow the same patch belong to the two views at the same time. In that ways, we can reduce this mutual informations, and we can just keep some task relevant information intact. And the other part is the cropping. Specifically, we use this to capture the regional heterogeneity, because the different regions, they usually will contain quite different diagnostic features, and we enforcing them to have some distance between these two different crops. In that ways, we can having a quite distinct, but all having a similar histological features. The last one is masking. The masking is mostly just for reduce the redundant visual tokens, because when you really like very nearby patch tokens, they may just look exactly the same. And also to put our all the patch token into the deep learning framework, usually you need a very good batching techniques to make sure like all every time the batch size we have is the same. So with masking, we can enable this efficient batch training. Here is the general views of using SPTs on these two different of generation of these two different views on the same whole set images with utilizing splitting, masking, and also the cropping. Here we have the red is in one view and the blue is in the other views. And after enabling the splitting, we will not have the same patch in both views. Okay. The another thought is why we just use the slide level transformations. Like when we do the patch training, we do have the patch augmentations and the patch encoders. Actually, there are some prior work they do enabling both patch augmentations and sliding augmentation at the same time. But we hypothesis like it's not necessary to these patch augmentations. Because actually, our SPD framework, at least, we rely on a patch encoder that is already trained in this training encoder that is invariant to different patch augmentations, which is usually how the patch encoder is trained. So if we visualize the patch tokens with their different augmented views on the t-SNE plot in their embeddings, you can usually find the anchor image of the original patches and their augmented views. They are very similar to each other's, and we don't think it will actually allow you to learn extra information about these slide representations. And actually enabling the patch augmentations, you may have to also have the inference during in this training for the patch tokens. However, if you just freeze them and extract all the embeddings together, you can have a more efficient training because you don't need the forward pass for your patch encoder. You just need do one forward pass and save them. That's it. We also compare our method with prior work. They also utilizing the patch augmentations, but they don't use the three transformations in the SPT, and we actually show that SPT is outperforming this baselines using its two-stage augmentations. Okay, so for the evaluation benchmarks here, we have 5 different benchmarks and we have 4 different tumor types. There are 2 image modality, both SRH and H&E, and on 3 different classes. For the SRH, we'll do the histological classifications. There are 7 classes. And for the H&E glioma, we'll continue benchmarking on this molecular classification of IDH wild type and mutant. For TCGA BRCA is breast cancer. We'll do a subtyping with 2 major breast carcinoma subtypings in TCGA BRCA project. Same as NSLC is lung cancer and RCC the kidney tumor. Okay. Here's the result for self-supervised SPT. The Giga SSL in the row above SPT is the one I mentioned about, like using both patch level augmentation and also the slide transformation, but just the masking. And it's showing that actually the SSSPT will outperform Giga SSL by a large margin. And also with the HIPD is another giant hierarchical transformer, and it's showing like the state of the art result, but we can still showing the SSPT having can outperform all those self-supervised baseline. On top of that, since we have another variant of the SPT called supervised SPT or SUSPT, we also benchmark SUSPT on all the state-of-the-art method in a supervised fashion. For example, TransML, DSML, and CLAM, all those methods. And actually, we can say in the self-supervised method, the SSSPT can beat all the supervised one. And for the supervised method, we also beat our baselines as well here on all different tasks. Here, we also want to have an ablation study of assessing the impact of different patch encoder. Here is more like to discover the relationship between the patch encoder and slide encoders. Because usually we will have like we may have a high disk patch encoder, it's pretty strong, but it's possible you don't have a very good patch encoder. Then how our SPD can work on this very like just ImageNet pre-trained network if you don't have the compute to train your own patch encoder. And also for some of the large lab, they may be able to generate a foundation model for a patch encoder. It's like it's a very giant transformer and put tons of data on it. It's like PLEP and Uni. And by utilizing the SPT, we can further boost their performance on top of their already extraordinary performance. Here, we classify the patch encoder into 3 different parts for the H&E benchmarks. We have out of distribution, which is patch encoder trained just on the ImageNet. We'll have the near or the in-domain patch encoder like HIPT, and we trained with SimClear, we train with high disk. They're all the patch encoder trained on the H&E data. And also the last 2, the foundation model, PLEP and Uni. PLEP is a vision language model. They trained on all the pathology images on Twitter on Twitter. They retrieve the text image pair and also the PubMed, some literatures, you can find the caption and the figures they describing this text and image pair. And the Uni, they use a foundation. They have their own in-house data set, which have like 100,000 whole slide images, covering like lots of tumor type, and they train a massive amount of data on this giant encoder. And we can say like truly the baselines here as a pooling method, we can say it's truly showing roughly showing the increasing performance as you have more and more strong patch encoder, and SSPT and SUSPT can basically diminish the difference of all your different patch encoder and leverage them to the same level. And if you're using an SUSPT, you can further increase the performance. This is another this is the same ablation study, just for another benchmark, for the SRH benchmark. Here, some of the methods, for example, like HIPT, which was trained on H&E data, now becomes out of distribution for the SRH data, because they are actually quite different. And the PLEP and the Uni, these 2 foundational models, actually will show there's a distribution shift between these 2 modalities. And the model like PLEP is just not working so well compared to the H&E benchmarks. And for the SimClear and high disk, these 2 methods, we train the patch encoders on in the SRH data. So they are basically in domain, and they just with the baselines, they are comparable with the Uni, and with SUSPT and SSPT, we can also boost their performance to the similar level. Like even you have quite different patch encoders. Yeah. As Uni claimed, is a state-of-the-art method just with using the ABML. Is a very easy and naive way of doing the feature aggregation on the patch tokens. But with using the supervised SPD, we can further improve their performance on a very high quality patch tokens. Here is the attention maps for all the patches we extracted from the host site images, and they really shows without any supervision. This is a entirely self-supervised method, and we can basically showing basically close to a unsupervised segmentation task, where you can see the clear tumor margins from the dense tumor and to the blood cell, and also here and some necrosis. They also have different attention values to look at. Basically the yellow part is with a high attention value. They're all showing the dense tumor. Okay. So SPD is a general framework for the whole slide representation learning. We show them on different benchmarks and image modality, and also SPD uses domain-informed vision language transformation for this whole slide image view generation, which is which enables this efficient training and also learns high quality features. Also, SPT achieved its superior performance on all the tasks we are showing here. Okay, so we have our general conclusion for our work. Here, we propose this training framework is quite general to basically all the whole slide images modality, whatever image type you are having. If you're having like gigapixel whole slide images, you can just follow this framework to patch them into smaller patches. This patch you can choose like either 20x or 40x. You can patch at 512 or 256, depends on how granularity and how powerful your computers have. And after that, we'll throw them into patch encoder. The patch encoder can be trained in a self-supervised way. We can use either just the conventional SSL, or we can also use high disk to leverage these hierarchical discriminations. After we get very high quality patch encoders, or we don't, we can use a slide encoder to aggregate these patch tokens into a single slide features. And here we propose this high disk and SPT to fill in these two rows here. But there are lots of downstream tasks for this framework, and also like, for example, here we are showing a tumor infiltration detection task, which is just to detecting the tumor margin of the glioma case. Here we can see the tumor infiltration from high to low on this whole slide images, and we're giving infiltration score, and this is a work we recently is accepted in the Nature. Here is the code GitHub repo for the high disk and SPT. You can have a look. Finally, I want to thank our co-author and collaborators in our machine learning neurosurgery lab at the University of Michigan. I would also like to extend our gratitude to all our sponsors. And any questions?
Any questions about either high disk or SPT? We'll be happy to answer.
I'll have a critical scenario. What will happen if you skip the patch encoder step and jump from the whole slide images to slide encoder? Would you get equivalent results or worse?
Yeah, I think that depends on, first, do you have a good GPU? You can actually feed in like entire raw image tokens into your slide encoder. Because here, our slide encoders actually is handling on the embeddings. This embeddings like 2048 or 512. However, in the image space, like after retiring them is an image is like it's 256, 3 by 256 by 256. So usually you still need like at least a ImageNet pre-trained encoder to encode them in embedding space. Because directly train them just in the imaging space is possible, but I think the easier way to separate two rows. One is to do a regional level learning for the patch encoder, and also aggregate them to form a better slide representation. There are some recent methods. They actually train them end to end. After the patch encoder training, they allow you to fine tune the patch encoder together with your slide encoder to do the aggregations.
All right, there was actually, I mean, you already showed like different applications to different tumors and TCGA, so I don't know if you wanted to expand on that, kind of the further applicability beyond gliomas.
Actually, we here showed more than just glioma for our baselines. Yeah, for the all different tasks. Here on the TCG, we have glioma and we also have the breast and the lung and kidney, so.
Yeah, but in the breast cancer, it didn't always do as well. I was just curious about that.
I would say breast cancer, this data set is pretty challenging. Here we are actually using, it's a very highly unbalanced data set. Even the state-of-the-art method, for the supervised part, they can achieve like around 80-ish method, and we can further improve them to 84. I would say is really hard to beat those baselines, and the metric we are choosing is mean class accuracy is the most truthful way to represent the performance on this highly unbalanced data set. Yeah, I think it's like 1 to 10 in terms of the.
Yeah, no,
Yeah.
that's really, thank you. And then my last question is like, how feasible this would be? For example, like, I know like those, the red 3 columns are H&E. But for example, for the SRH, like how feasible is it actually to like, for, for like, I guess, maybe, can I say, like lower resource hospitals to have an SRH imaging machine, there, if you have an idea.
I think
Yeah.
about it. It's actually cheaper to have an SRH machine compared to having a full lab and have the technicians have staff to maintain the H&E lab. So H&E is certainly more popular in a lot of places, and that's just because that's been the standard for the last 100 years. But if you actually compare the cost of acquiring acquiring an SRH device and maintaining a lab for doing H&E staining and all that stuff, the SRH device is probably cheaper. Like you don't need a lot of training to use SRH, like I can do it, so there's that. So I think going forward, I think both modalities will be important and quite interesting for how basically the digital pathology community moves forward, right? Because I don't think just doing H&E is going to be the future of how we do these diagnoses.
So, yeah, or.
I'll talk.
I don't know much about histology. I was completely confused this entire time. But it's more like related to so I wanted to ask, you seem to be like a very knowledgeable histology, and I wanted to ask like so the standard is identifying tissue abnormalities of body histological statements. Pathologists look at the statements and determine like the abnormalities. But what do you think about the possibility that these abnormalities can also be identified on the molecular level before the physiological, like before it manifests something pathological?
Before something pathological. So that's very interesting question, but I don't think that's something that we're particularly looking at. I think a lot of the ways that we're using these images, or what happens is that some patient comes in to see their physician, maybe they have a headache or have something, and then they will probably get an MRI scan then. And then maybe they see a tumor or maybe they see something else. So kind of at that point is where a lot of these diagnosis tools can come and help to do these diagnoses, and where like maybe physicians can intervene during surgery or the biopsy. But before that, like if you're talking just about like genetic screening or something like that, I think that's something that's possible, but you probably wouldn't have any images to do these screening. You probably just do some sequencing data, which you can probably do a lot of data mining on that as well, but that's something that I'm less familiar with.
If I may, just a quick question, Mr. Chen, I know you are always an advocate of cheap computational or like a computational efficiency. So my question for you and Renly is how much computational resource you have to use in order to run your pipeline in terms of time or like a GPU wise?
I think I can answer your question with our oblations on patch encoder. So as I mentioned, this is two-stage training. We have our patch training. We also have slide training. Potentially, we also have a patching stage. The patching stage, I think, is pretty fast. This part, you don't need to worry too much. You just patch name and you just filtering on the like some intensity, bluff, and you filter background.
CPU.
Yeah, so that is all CPU is very easy. So the bottlenecking part is the patch encoder and the slide encoder. Most recently, I think most of the patch encoder, they are the very heavy part to train. So for the like image, we don't talk about ImageNet because it's already off the shelf. But the high disk sim clear and high disk, if you want to train in-house, currently we are using two L40 GPU. They have like 48-ish GPU memory, and then we train it for 2 days.
At least.
Yeah, roughly. And for the Plep and the Uni, those are very large model. As for the Uni, as far as I know, is they are using 48-800 GPU and train a week. Yeah, so
They train these models for weeks, and then you also need to kind of factor in tuning and things like that.
yeah.
Right.
Yeah.
But the good news for us is that ImageNet, Clip, Uni, if you email me high disk, you can get these models for free, right? Essentially.
Yeah.
So like that's kind of like the heaviest part of training. If you want a really, really good one, it's probably gonna take weeks and months to train. So there's that. And then the sliding coders are faster. So all of our experiments, the sliding coder just took one L40 or A40, this one we have on the at ARC, and we basically took them it took it like a few hours to train.
6 to 8. And here also, this is model size for the data size. This in-domain data set was just specifically training on the data like we are going to evaluate. For example, we are going to evaluate breast cancer. We just train a patch encoder that is just for breast. And for this foundational patch encoder, they show their extraordinary generalizability on all like different tumor types you can think of. You can find on like online, like 30-ish tumor type. They can always showing generally okay, not the best, but it's usually easier for you to train some data from by yourself. Like just evaluation here with a baseline. Their generalizability on this H&E glioma is just better if you just train your own patch encoder. So some of time you can just utilize their methods, and on top of that, you can use SPT to further boost performance on the task you are interested. And this part is light. Yeah, and this part is just one GPU, 6 to 8 hours.
That's a good place to end, so we can thank Cheng and Renly one more time.